Okay, good afternoon, everybody. And here's our, this is going to be our only class this week because Wednesday you have a test. And the test will take place during the class time, during the set class time. We'll take up the whole class period. Not the whole class period, sorry, it will take up an hour of the class period. So we won't have another class on Wednesday. Today we're going to continue our discussion about the structure and function of large molecules in the cell. Last time we discussed polysaccharides and lipids. And today we're going to discuss the proteins and nucleic acids. And of course, the proteins, nucleic acids are intimately connected with one another because the DNA of the cell, which is a nucleic acid, contains the recipe for all of the proteins in the cell, and much of the functioning of the cell is driven by proteins, as we'll see in a second. So the proteins are an incredibly diverse group of molecules. They share common features, as we'll hear, but they, first of all, they account for a large, substantial part of the mass of the cell. If we dry a cell and examine it, we'll find more than 50% of the cell is actually composed of proteins. They are very, very important structural elements, but they also have a wide range of functions within the cell. They may be for storage, transport, all sorts of things, as we'll hear. But probably their central role that we need to think about is their function as enzymes in the cell. We will discuss this in more detail when we talk about the cell metabolism. But most reactions inside the cell would not occur spontaneously at any rate which would be useful to the cell. They would, under normal circumstances, most biological reactions are complex reactions, which would only take place very slowly if they happen spontaneously at all. Instead, the reactions inside the cell, the reactions of metabolism, are shepherded by proteins. And these are the enzymatic proteins, as shown here. Now, the enzymatic proteins, they accelerate the rate at which reactions occur and make them biologically useful. Without the enzymes, they would just take place so slowly, they would not be biologically useful. So they selectively accelerate chemical reactions. The enzymes are extremely specific to the reactions that they cause. In fact, they are very specific to the substrates that they bind. Typically, for example, a catabolic reaction might take a disaccharide, as shown here, and break that disaccharide into its component monosaccharides. That would be a typical enzymatic reaction. We'll hear a lot more about enzymes later on. Remember, 99% of the enzymes in the cell are proteins. There's one exception, and that is that some nucleic acids can act as enzymes. So there are some other substances which can act as enzymes, but enzymatic activity in the cell is usually by means of proteins. Second of all, proteins often play a defensive role. And the classic example for this, protection against disease, for example, antibodies, which we hear so much about nowadays, especially as we deal with the COVID-19 crisis. We hear about people developing immunity, etc. That is done by the immune system, and the immune system manufactures antibody, which is a protein, which is specific to a particular part, usually, of some pathogen. And the antibody is able to bind the pathogen and inactivate it. It can also be activated against toxins and various other things. Antibody is extremely powerful, highly, highly, highly specific product of the immune system. Next, proteins may be used for storage. This is not always a common thing, but they can be used for storage. The classic example of that is with egg white, for example. Albumin is a protein, and very often eggs ova of many animals contain abundant albumin as a storage product, as a source of nutrient and energy. So in eggs, the albumin it's actually called ovalbumin, and it provides amino acids for the cell. Next, all of the functions of transport at the membrane and also within the cell are the responsibility of proteins. And so, for example, hemoglobin. Hemoglobin is the protein which is present in blood cells, and it transports oxygen around the body. That's just one. Second of all, within the membrane, embedded into the cell membrane, embedded into the plasma membranes of the organelles, there are often proteins which are highly specific for transporting substances to and across the membrane. It's the proteins embedded in the cell membrane, for example, which allow the cytoplasm to be different to what the outside medium. It's the proteins which selectively transport substances either way through the membrane. That is one of the major transport functions of proteins. Proteins do also function as hormones. There are a number of proteinaceous hormones, and a classic example there is insulin. Insulin is produced by the pancreas, and it's a hormone which governs the uptake of glucose from the blood into the cells. If your blood sugar rises, insulin is released, and it stimulates the cells, particularly of the liver, to take up glucose and drop the concentration of glucose in the blood. So insulin is a proteinaceous hormone, but there are other protein hormones as well. The very, very, very important function of proteins is to act as receptors. And to explain this, let me just repeat what I've said before. A single cell on its own has no eyes, it has no ears, it has no way to, yet for all that it's blind and deaf and everything else, it needs to know what's happening in its environment. It needs to know especially what the environment contains in terms of nutrient, the pH, all of that sort of thing. It is the responsibility of proteins embedded in the membrane to do that sensing, to sense what is happening and to relay information to the interior of the cell. Those proteins that do that kind of job are called receptor proteins. And here's a classic example of a receptor protein. These are receptor proteins embedded in the membrane. Let's just say of a muscle cell, okay? And when that muscle cell is signaled to contract, for example, a nerve impulse is generated, and the nerve impulse reaches the end of the nerve next to the muscle. That nerve cell will release substances called neurotransmitters, and those neurotransmitters are picked up by receptor proteins. They actually bind to the receptor proteins, and those receptor proteins then convey a signal. They don't transport the substance across. Instead, they generate a signal to the cell. In a muscle cell, they generate a signal to cause it to contract. But there may be other ways in which receptor proteins work. There may be other substances, for example, that they bind. The receptor proteins are extremely important because they are the cell's chief means of communicating information from outside to the inside of the cell, and also within the cell as well. Next, all movement in the cell and by the cell arises because of these proteins, the contractile and motor proteins. And it doesn't matter what kind of movement you're talking about. Well, let's think, for example, about the movement of an amoeba, let's say. You remember that amoebae are single-celled. They creep around on the substrate by extending out what are called pseudopodia. They extend by changing the shape of the cell continuously. They can move fairly rapidly, and all of that movement arises because the cell has a cytoskeleton. You'll remember we referred to it before. And the movement arises because that proteinaceous cytoskeleton is continually broken down and reconstructed to change the shape. That's one sort of movement that arises because of proteins. But there is a whole other area of movement which involves the interaction of proteinaceous filaments and a group of proteins called the motor proteins. And we'll discuss it in more detail when we, for example, talk about muscle activity. But the things like muscle contraction are caused because protein filaments, contractile filaments, slide against one another. And they slide against one another under the action of motor proteins. In addition, movement of stuff within the cell, transport of vesicles, transport of organelles, movement of organelles from one part to another. All of these kinds of movements are also the responsibility of proteins, and they are usually because proteins called the motor proteins interact with other protein filaments. So you can think of it, for example, I want to send a vesicle from one part of the cell to another. The cell will build a proteinaceous filament, like a railroad track, and a motor protein will grab the vesicle, or whatever it is we want to move, and it will then march along that filament, dragging the vesicle with it. So all of these sorts of movement are because of contractile and motor protein. Next, we've said like 50% of the cell of the dry mass arises because the cell has so much protein, and much of that protein is structural protein. It is protein which builds the structure of the cell. It gives it its shape, and it also allows the cell to adopt a particular shape for a certain length of time and then change it. If we change that skeleton, we can change the shape of it. There are other proteins. A simple example of another structural protein is that of keratin. Keratin is the protein which makes up our fingernails and our hair, for example. In other animals, there are other structural proteins, silk in spiders, for example, and in caterpillars, which weave their cocoons. Those are all structural proteins. And in tissues themselves, quite apart from within cells, but within whole tissues, there may be extensive networks of protein laid down. For example, in cartilage, tendons, all these things, there are proteins, in particular one called collagen, which give tremendous strength to the connective tissues. Okay, so those are just some of the functions of proteins. Pay careful attention to the assignment that I set that will cover this next week, because in there will be some discussion about other functions of proteins that you might need to know. Okay, so let's have a look at this, actually how proteins are made and how it is that we end up with this huge diversity. One thing I need to emphasize right at the very beginning, protein function is intimately concerned with its structure, and for reasons that I will explain. But in a particular protein has a particular function because of its structure. If I change that structure even slightly, I will run the danger of radically altering the function of the protein. As we explain how proteins are structured, you'll begin to understand why. First of all proteins are basically polypeptides. They are a polymer. And the monomer that makes up proteins is called the amino acid. So proteins are polypeptides. That means there are many amino groups joined together. And those groups are joined together by what is called a peptide bond. That is where the word polypeptide comes from. They fundamentally start off as a chain of amino acids all joined together. That is the polypeptide. But that polypeptide often becomes extensively modified. That, for example, was the function of the endoplasmic reticulum, the rough endoplasmic reticulum especially, and the Golgi, is to take polypeptides and modify them. And when they are highly modified and reach their final functional form, that is what a protein is. A protein is the functional form. It's the functional molecule formed from one or more polypeptides. So the size of protein, there are some small proteins in the cell, but most of the proteins are really, really big. They can be enormous molecules. They are very strong molecules because the polymer is built from covalently joined groups, covalently joined monomers. So here's, you must remember this structure. This is the basic structure of the amino acid. All amino acids have the same basic structure. And that is why we can join any amino acid to any other amino acid. They can be, any amino acid can join any other. We can make any order necessary to construct the protein because they all share this. That's a classic rule of polymer formation. So here, have a look here and you'll see. This here is a central carbon called the alpha carbon there. And attached to the alpha carbon, there are two groups. There's an amino group. That's where the word amino acid comes from. The amino part of the amino acid comes from that. And then there is an acid group, a carboxyl group there. That's where the acid part, amino acid, amino acid. And there are 20 different amino acids that are used in cells. There's more amino acids than that, by the way, but there are 20 used in cells to build polypeptides. And each individual amino acid differs from the others because it has on the one side a side chain called the R group, sometimes referred to as the reactive group. You can just remember it as the R group. It's on a side chain. This side chain does not participate in polypeptide formation. It has nothing to do with making a polypeptide. It has nothing to do with forming a polymer. The polymer is formed by joining the base group that forms the structure of the amino acid. So have a look here and we'll see, you'll begin to get an idea now about the different amino acids and different things that they can do. They differ from one another tremendously. An individual amino acid, one individual amino acid may have a completely different character to all of the others. And that character is given to it by the side chain. Here, have a look here, and what have we got? We've got, here's our central carbon here. Here's our amino group. Here's our carboxyl group. Same here. Central carbon, the alpha carbon. Here's the amino group. Here's the carboxyl, etc., etc. They're all identical. But when you look at their side chains, you'll see the side chains vary tremendously. Here's just a single hydrogen, methyl group. Now we begin to have a look at these. What do these start to look like? As you look at them, lots of carbons, lots of hydrogens. They begin to look like hydrocarbons. And that is indeed exactly what they are. They are like lipid groups attached to the side of this central group that we can make the polypeptide from. All of these particular amino acids, you do not have to memorize the individual properties associated with the name. But just take note, all of these side groups here are non-polar, like hydrocarbons. They are non-polar and strongly hydrophobic. So just imagine, in your mind, just imagine that I make a polypeptide. I make it of all sorts of amino acids, just long chain. And then suddenly in the middle of that chain, I put a whole lot of non-polar groups, amino acids with non-polar groups. That area where I've packed full of non-polar, how do you think it's going to behave when I put that protein into water? What is going to happen is that all of these non-polar groups are going to be repelled, and it's going to force that molecule to start folding away, folding those groups away from the water because they're strongly hydrophobic. This is a way in which accumulation of particular side groups in an area of the polypeptide can begin to influence both the structure and then the function of the protein. That is what is going to happen. We're going to take specific groups and we're going to put them into a particular area, and they're going to start interacting chemically with the environment to do particular jobs. That is how proteins work. So this is the whole group with non-polar side chains, but there are others with highly polar side chains. These are strongly hydrophilic. Okay, you can see here, look, OH, OH, OH. This is a minor group and all sorts of groups. These here are polar, strongly polar. They are highly soluble. So imagine that I put highly soluble, you remember we just talked about having a region that was hydrophobic. How about I flank that with hydrophilic? What is going to happen? They're going to dissolve in and they're going to zip that hydrophobic area. This is how we start to, this is how the molecule, the polypeptide, begins to acquire structure. And if it's acquiring structure, it's acquiring function. So we've had non-polar groups, polar groups. Here we have strongly electrically charged groups. These here are acidic. They are negatively charged. These are basic groups. You can see, especially because they've got lots of nitrogen. As soon as you see nitrogen, you know that it's probably going to be basic. So these are strongly basic. We've got polar charge, non-polar. We've got polar character, non-polar. We've got a negative charge, positive charge, etc., etc. We can begin now to think that if we can only get these side groups into position, they may be able to interact with molecules in their environment. And that's exactly what happens with proteins. So if I want to make a polypeptide out of individual amino acids, then what happens is we have to perform a dehydration reaction. We heard about that before. Polymer is made by a dehydration reaction which takes out a water molecule. So have a look here. These are two amino acids which are already joined together. There's one and there's the other, and they are joined together here. You can see between this carbon and this nitrogen. So it's this carbon of a former carboxyl group, this nitrogen of a former amino group. So here I'm bringing in another amino acid here, and I want to join it to this little chain. And this would be called a dipeptide because it's only got two. So now we're going to join this to there. We do it. Have a look here. Here's the alpha carbon. Here's the amino group. Here's the carboxyl group. Here on this chain here, the only reactive group that I have available is this carboxyl group here. There's an H, there's an OH. We take those out as water, and what we end up with is forming a bond between that carbon and this nitrogen. this carbon and this nitrogen, that carboxyl group is now free and available for another another amino acid to be added to it if need be. We formed a bond between the carbon and the nitrogen, and that is called a peptide bond. It is a peptide bond. And you'll notice that the amino acid, this little short polypeptide, is not symmetrical. It has an amino group there on that end, and it has a carboxyl group there. And whenever we add another amino acid, we're going to add it here. We can't add it there. We've got to add it here. So the chain will grow in that direction there if it grows at all. So realize that looking at this, that because these groups are all the same, we can in fact add any amino acid to any other. And the order of the amino acids in a polypeptide is critically important. And that order of amino acids in the polypeptide is determined by the gene. That is what the gene does. The gene has the recipe for the polypeptide. So proteins assume very, very complicated three-dimensional shapes in space. And as I've told you, their three-dimensional shape in space is actually what determines their function. So have a look here, for example. This is the interaction of an antibody with a protein from a virus. This is actually a visualization of a vaccine, of antibody which has been produced in response to a vaccine. And the vaccine was that was introduced to the person was in fact protein, a small protein from influenza virus. Here it is here. Look at its shape. It has this very, very intricate three-dimensional shape. They've shown this here as a contour surface to give you an idea of the complexity. The body has produced an antibody in response to the presence of this protein. And look at the antibody. Just look at the matching of the shape of the antibody. It is absolutely faithful to the shape here. It is remarkable. This has to be like this. Antibody is extremely specific. It is extremely specific to the substance that it has been produced against. And it acquires its specificity because, first of all, because of the shape here that surface adopts, but also because of the reactivity of the different side groups that are now exposed here and can interact with this protein. And what happens in the body is that these two bind together. And if this protein was attached to a virus, for example, all of the antibody binding to this protein on the surface of the virus would inactivate it and stop it from being able to infect cells. That is the idea of, that is how this vaccine actually works and how the antibody works. So we need to ask the question then, how is it that the protein acquires its three-dimensional shape? Well, there are actually, we think of 4 levels of protein organization, each equally important. The first we could refer to as the primary structure. Primary structure is simply the linear sequence of amino acids, which is determined by the gene that governs the production of that particular polypeptide. So the primary structure is simply the linear sequence of amino acids. When we do that, remember that each amino acid has the same base structure, right? There are certain features of those base structures. For instance, areas of charge, slight polarities, things like this, which begin to interact. So those charge and things interact with amino acids that are close by. And they can induce one of three things can result from that. The first is regular repetition of particular charges, influenced by the side groups as well, will induce the formation of helices in the primary structure. So now our primary strand begins to fold, and it can fold into helical structures. It can also fold into flat structures. It depends on what the nature of those chemical interactions is, into flat structures or into none. If it has none, then the primary structure is simply loose and the strand is easily bent. So those are useful areas because they act like hinges. We can have, that's the three features of the secondary structure: alpha helices, beta sheets or beta pleated sheets, and then nothing, a hinge area which doesn't have any secondary structure. And remember, those are all formed because of repeating constituents of the backbone that start to interact with one another. Tertiary structure is structure which is induced between various side groups of the amino acids, but over a bigger distance. So in other words, we can take areas of the polypeptide which are quite remote from one another and get them to interact. And that induces tertiary structure. There are various things, various ways in which molecules can begin to interact with one another. The first, very frequent, is by hydrogen bonding. If we have molecules which are polar, then we can have weak hydrogen bonds established, which can begin to fold and bend the molecule. There may be ionic bonds as well. Remember that we have electrically charged groups, for example, which can begin to establish ionic bonds. There are other kinds of interactions which you may not have heard of: hydrophobic interactions, van der Waal interactions. Just be aware that these are weak interactions, but can be very important in establishing tertiary structure. There's one which is incredibly important and quite frequent, and this is the formation actually of covalent bonds between different areas of the molecule. And this arises because of one particular amino acid called cysteine. And cysteine has a sulfur group attached to it. And that when you have two cysteines that can be brought close to one another, they can interact and they form a covalent bond between the sulfurs. Sulfur to sulfur can be joined with a covalent bond. And that is called a disulfide bridge. Very important because it's such a strong, that is such a strong bond. It's a covalent bond. All of these other bonds here, very important in establishing tertiary structure, but they are weak. They're relatively weak. They may be very frequent, so that can be actually, add up to a lot. But the disulfide bridges are extremely strong and make a permanent bond, if you want to think about it that way. Now, there is a fourth level of structure, which does not apply to all proteins, but many proteins are actually, the mature protein is formed because two polypeptide chains interact with one another, or more, two or more interact with one another to form the mature protein. Hemoglobin is a good example. Hemoglobin arises because of interaction between four molecules of hemoglobin, 2 alpha and 2 beta. They interact to form the mature hemoglobin molecule. That we call quaternary. And that again that quaternary structure, the links between the proteins can be by covalent or non-covalent bonding. Very often it's just by hydrogen bonding, things like that. But you can have disulfide bridges that stabilize the quaternary structure. So if you think about, we've talked already, collagen, for example, is another protein with quaternary structure. It's 3 polypeptides that are wrapped around one another to form these very strong fibrils. Hemoglobin, I've already told you, it's 4 polypeptides, 2 alpha and 2 beta. And there are many others that have quaternary structure. Be very careful that you memorize these. Go to your textbook and have a look for a bit more detail on how these are structured. And I will also put up a series of small animations. I'm not going to do them here in class. Small animations which may help you to understand the difference between primary, secondary, tertiary, and quaternary structure. Classic thing to ask questions about in an exam, by the way, but also very important that you just understand it anyway. Okay, here's the primary structure. This linear sequence of amino acids, and that is determined by the gene. The other elements of structure are determined by interaction between groups. Secondary structure, but interactions between quite closely linked groups, like so they lie close to one another, pull the primary strand into either alpha helices or into beta pleated sheets like this. And then there may be some short areas which have no secondary structure that acts like hinges. And what can happen is we can begin to bend these alpha sheets, alpha coils and the beta pleated sheets. We can begin to bend them in space, bring them close to one another, fold that molecule, and start to create a 3-dimensional shape in space, as shown here. This is the tertiary structure, and it is established because of bonding between remoter areas of the molecule, which pull the molecule into its 3-dimensional shape. By various bonding techniques, may be stabilized by covalent bonding, but a lot of this folding is due to things like hydrogen bonding. Then the quaternary structure, we take 2 or more polypeptides and we join them together. Here, by the way, these are very, very similar proteins which are in form the quaternary structure. There's sometimes the polypeptides may be quite different to one another in the mature protein. Primary, secondary, tertiary, and quaternary. When you have a look at diagrams of proteins, you'll see that they're often very, very large, extremely complex, and biochemists use various ways to try to describe them graphically to understand their function. And here is one. This is an enzyme called lysozyme. And lysozyme, it's naturally produced. We have it in our tears, and it attacks the cell wall of bacteria. It's very specific in the way it does it. It only attacks very, very specific groups of chemicals in the cell wall of bacteria. But it's very good at what it does. It's a good antibiotic, naturally produced in tears especially. So have a look at its structure here. What have we got here? We've got an alpha helix. Okay. Another alpha helix here, some loose hinge areas. Here's a beta sheet here. There. The whole thing intricately folded in and back on itself in its tertiary structure. So here's its primary structure, just the sequence. Secondary, alpha helices and beta sheets. Tertiary structure, all of these folded into a 3-dimensional shape by various bondings. And what it's done, it has created a groove here. And concentrated in that groove are the side groups of various amino acids whose interaction does something. Their interaction actually will break down a particular molecule called peptidoglycan, which forms the cell wall of bacteria. And that activity is limited to this little area here. And we call that the active site. And all of the rest of the protein, all of the structure is there so that we can construct this very, very specific groove with its specific geography and specific side groups in that groove, which are going to do one particular chemical job. So here is a space filling model. It's just to give you an idea of what it would look like if we could zoom down and see it in three-dimensional shape. Here is the groove here, and this would be the active site of this particular, this is an enzyme, lysozyme. So those are all the determinants locked into the molecule of that determine the protein shape and everything else. But we need to realize also that many of those things that we talked about, hydrogen bonds, the ionic bonding, even van der Waals forces, all of these sorts of things can be very strongly influenced by the environment. And the proteins can be radically changed if we change pH, the acidity or alkalinity of the environment, if we change the salt concentration, if we change the temperature. All of these things will affect protein shape. And we find that proteins have an optimum. And we especially see this with the enzymes. The enzymes have a very, very specific chemical function. And they have an optimum environment in which they operate. And that optimum environment is the environment which most closely preserves their shape and function. And under normal circumstances, for most cells, the environment is more or less neutral. In our cells, it's mostly relatively neutral. If I radically alter the environment, if I have a protein and I radically change its environment, say I change the pH, or I radically change the temperature and things like this, I will affect the actual physical structure of the protein, because I'm going to start interfering with the bonding that establishes secondary, tertiary, and quaternary structure. Many of those bonds are relatively weak and very easily disrupted. And so we find for a particular protein, it has an optimum environment at which it operates best. And if we deviate from that, we run the risk of actually denaturing the protein. We disrupt the structure so much that it stops functioning. Now, in certain environments, we can actually restore the protein. We can renature it. But very often denaturation is very dangerous because the proteins do not restore. They cannot restore their function. If you take egg white, for example, egg white is it's a liquid, but it's almost a gel because the molecules are so enormous that makes that kind of slimy jelly kind of consistency. But if you heat egg white, what happens to it? It solidifies. And the reason of that is because it loses its normal natural bonding, and it establishes very strong covalent bonds between areas of the molecule that are not normally associated with one another. Well, you can cool egg white down and you're not going to restore its function. And the same thing applies to many proteins. If you denature them too radically, they will actually die. They will lose their ability to function at all. Others can be restored. And I just want to mention something as well. You know, there are organisms which live under very, very harsh conditions. So, for example, we have bacteria that live in very, very acidic conditions. There are bacteria which live under very, very hot temperatures, above boiling point in deep oceans, for example. And they are able to do that because they have specialized proteins. Their proteins are adapted for those harsh environments. And if we were to take those organisms and put them into a normal neutral environment, their proteins would also denature. Their proteins are adapted to those environments. The proteins are very, very versatile. And we can find proteins that function under extraordinary circumstances. Okay, so please do read through that chapter. I've skimmed through. I don't need you to know a lot more detail than I've given you, but do please read through that and get an idea about some of the stuff maybe that I have not taught you about. Okay, so we've said the first important thing that we need to know about a protein is that primary sequence, the sequence of amino acids that may give the protein, the polypeptide, its primary structure. That is determined by genes of the organism. The recipe for the primary structure is contained in a gene, and for the most part, we can think that one gene leads eventually to one polypeptide. The genes, the hereditary material in our cells, is made of DNA, deoxyribonucleic acid. That is a nucleic acid, and the nucleic acids are a class whose properties we're going to discuss now. The nucleic acids are also a polymer, but the polymer of nucleic acids is made up of monomers, which are called the nucleotides, and there are only 5 of them. Despite the fact that DNA, for example, has to contain all the recipes for all the proteins in the cell, we are able to include all of that information in DNA, despite the fact there are only 5 nucleotides, because again, we can join any nucleotide to any other nucleotide, because all nucleotides have all the nucleotides in a particular nucleic acid have a base structure which is identical, through which the joining will take place. The character is given by another part of the molecule, but the joining is through a unit, a root, which is identical in all nucleotides for a particular nucleic acid. So the hereditary material is deoxyribonucleic acid, DNA. You'll see what characterizes it in a second. But unfortunately, the DNA cannot talk to the cell. It cannot talk directly to the cytoplasm. The cytoplasm just doesn't talk DNA. Instead, the recipes of DNA have to be transcribed, and they are transcribed into this molecule here, ribonucleic acid. Now, ribonucleic acid, or RNA, differs from DNA in very significant ways, but there's only one kind of DNA that we need to worry about in the cell, and that contains the hereditary information. But there are many kinds of RNA. And the very first one we're going to meet is called messenger RNA. And messenger RNA is a copy of DNA. It's not really a direct copy, it's a transcript of DNA, as you'll see when we discuss DNA function. So DNA is a fascinating molecule on its own. DNA, obviously, the cell divides, DNA needs to be replicated really accurately, and the DNA, just in its structure, contains all of the information necessary for its own replication. And no recipe has to be kept anywhere else of the DNA. The DNA contains recipe for its own replication. DNA also directs the synthesis of messenger RNA, and the messenger RNA is what goes out to the cytoplasm, and the elements in the cytoplasm, the ribosomes, are able to read the messenger RNA. And the ribosomes read the messenger RNA and produce the polypeptide, which goes on to form the protein. So that is the take-home message from this. We have DNA. The DNA is transcribed to messenger RNA, and the messenger RNA is then translated to protein by the ribosomes. And that you'll see it written down in a while. That we refer to as the central dogma. And it's a central dogma that DNA is transcribed to messenger RNA, which is translated to protein by the ribosomes, because that is the way all life works. From the simplest bacterium all the way up to the most complex multicellular organism, that is the way all cells work. That is the passage of information in the cell. And we'll refer to that very frequently again later. Today's class, all I'm going to do is cover some of the physical structure and some of the basic rules for DNA structure. When we talk later on in this course, we're going to talk about DNA function, and then you'll hear much more about how these structures that we're going to talk about today relate to the processes of replication and transcription and all sorts of other things. For today, just try to get the physical makeup of these nucleic acids clear in your head. So first of all, this diagram, by the way, contains basically everything that you'll ever need to know about the structure of nucleic acids locked into this diagram. I hope I can unlock it for you. Let's talk first of all about the unit structure, the monomer that makes up the nucleic acids, and that is called the nucleotide. And here is a nucleotide here. And this is a typical nucleotide. There are 5 nucleotides that we are going to talk about. All the nucleotides for a particular nucleic acid have a root structure, and it is this here, that there. And it's through these groups here that we're going to actually build up these enormous molecules that may result. So first of all, this is a sugar molecule. And remember what I told you about the sugar molecules, that they're drawn in shorthand. So that here at this point here, first of all, there's orient yourself. Here's an oxygen molecule. They're tying up this into a ring structure. Here's a carbon here. Here's a carbon here, 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 and here. There are 1, 2, 3, 4, 5 carbons. It's very important that you get used to the idea that we number those carbons. This is called the 1' carbon, 2' carbon, 3' carbon, 4' carbon, 5' carbon. And they have their various their importances, which I'll explain to you. The first thing I want to point out, there are in nucleic acids, there are 2 kinds of sugar. This is a pentose, a 5-carbon sugar, 1, 2, 3, 4, 5. It's a 5-carbon sugar. But there are 2 kinds of pentoses that we need to think about. The first is the pentose that is contained in DNA. Have a look here, and you'll see this group at the 2'. There's only a hydrogen there. Look at this one. This is ribose sugar. This is what we have in RNA. Have a look here. It has an OH. If I take away that O, I have deoxygenated it, and I end up with this group, this here, which is deoxyribose. This is ribose that has lost an oxygen. This is deoxyribose, and that is what is used in DNA. That is the functional sugar in DNA. In RNA, the 2' has a hydroxyl group there, and so it is ribose. And this great distinction between these two, and it's one of the ways in which the cell knows what it's dealing with. This is a label for the cell, so that the cell actually understands that what it's doing is it's dealing here with ribose, here it's dealing with deoxyribose. So those are up, and there's one of 2 possibilities depending on what nucleic acid we're talking about. Have a look here, and you'll see that at the 1', we can attach any one of 5 groups of nitrogen-rich ringed compounds. We call them the nitrogenous bases. They are very weakly basic because of the presence of all that nitrogen. And those ring structures consist either of single rings here or of double rings, as here. And the single rings, cytosine, thymine, and uracil, are called the pyrimidines. So single rings are pyrimidines. Double rings are purines, and they are adenine and guanine. So I'll tell you straight away that I said we made up our nucleic acids out of only 5, but in DNA, they only use 4. And in RNA, they only use 4. So in DNA will use cytosine, thymine, adenine, and guanine. But in RNA, there is no thymine. And instead, a very similar molecule is substituted, and that is uracil. So in RNA, as soon as you see uracil, you know you're dealing with RNA, right? So RNA has the following. It has adenine, guanine, cytosine, and uracil. We abbreviate those to CTUGAG, but please be sure that you know the full names. Be ready to produce those names in full, okay? It's very, very important. Do not just know C and not know what C stands for. Cytosine, thymine, uracil, adenine, guanine. Cytosine, thymine, adenine, guanine in DNA. Guanine, adenine, uracil, and cytosine in RNA. Another way for the cell to know what it is dealing with. Those groups are attached here at the 1' carbon. 1' carbon are ring structures. 2' carbon tells us, is this deoxyribose or ribose? It's either an H or an OH group. Here is the group that you really, here's the carbon site you really need to know about. The 3' carbon has attached to it an hydroxyl group. When you go to sleep at night, after you said your prayers and before you go to sleep, I want you to lay your head on your pillow and go 3' hydroxyl, 3' hydroxyl, 3' hydroxyl. If you understand and remember that the 3' hydroxyl is the only place on this nucleotide where a reaction can take place, you'll be 3/4 of the way to understanding the function of DNA. There's only one reactive group on a polynucleotide. When I start joining these all together, there's only one place on that polynucleotide where a reaction can take place, and it is here. It's at this 3' hydroxyl, that group there. If I have a chain like this, and I want to add another nucleotide, I can only add it here. I cannot add it there. It's impossible. I have to add it here. The 3' hydroxyl ends up being extremely important. Okay, extremely important. So we've got some basic things now. But the next thing is, don't worry about 4, there's this carbon there. All we need to know about that is that it has this group attached to it here. The 5th carbon is attached to the 4th, but attached to the 5' carbon is a very important group. It's a phosphate group there. And you'll see why that is so significant in a second. Okay. Here's our nucleotide base attached to carbon 1, carbon 2 hydroxyl or dihydroxyl, or H or OH group, 3' hydroxyl, 4', 5', the phosphate group. This you must know. Okay, this you must know. So the first thing to tell you is, let's just talk about DNA for the moment. Here, have a look here. This could be one strand of DNA. DNA is actually a double molecule and has 2 halves to it, but we'll just think about 1 half for the moment. 1 half of the DNA is constructed of very strong covalent bonds which form between that hydroxyl group of one nucleotide and the phosphate group of another. So when I look here, what I can see is here's my sugar, here's a phosphate, here's a sugar, here's a phosphate, here's a sugar, here's a phosphate, here's a sugar, here's a hydroxyl group ready to receive another nucleotide. And if I bring in another nucleotide here, I can react the phosphate group to the hydroxyl group through a dehydration reaction and form another very strong covalent bond. As a result, the DNA molecule, this side, has a very strong backbone. This is one of the reasons that DNA is such a durable molecule, is it has a strong backbone, and that backbone is quite difficult to break. The bonds are really strong. So when you hear about DNA being extracted from body fluids or something at a crime scene, et cetera, that may be very old, but the DNA survives, and it survives largely because of the strength of these covalent bonds in the backbone. So that is one strand. I just want to point something out to you, though. This strand is not the same at one end as it is at the other, because it only has one end that has the 3' hydroxyl. That's the 3' hydroxyl there. We call this the 3' end of the molecule. At the other end here, there is a phosphate there. That is the 5' end. So we have a phosphate at one end, hydroxyl at the other. Okay, but I can only react there. That's the only reactive site. But remember, this is very important because it lets you orient yourself. This molecule is not symmetrical. It has a 5' end and a 3' hydroxyl end there. But DNA is not a single molecule. RNA is. RNA looks like this. It's a single chain. But DNA is not. DNA is a double molecule. And I should just tell you quickly something about this early work on DNA. One of the very first things that was ever noticed about DNA was it didn't matter where you took your DNA from. If you took it from a human being or an anteater or an ant or a bird or whatever, the DNA might be very different in those different things. But when you analyze the DNA, what you discover was that in DNA, there is always as much cytosine as there is guanine. And there's always as much adenine as there is thymine. It's an invariable rule. If I've got X amount of cytosine, I must have the same amount of guanine. If I've got X amount of adenine, I must have the same amount of thymine. That is called Chargaff's rule. And it was one of the great mysteries of DNA as to why this was the case. But that mystery is solved when we look at the structure of the double molecule. And here we are here. So have a look here. Now take this strand here, it's oriented like the one we just saw, 5' to 3' there. 5' to 3'. And you'll see that these molecules form this lovely lazy spiral in space. Every time you join a phosphate group to the hydroxyl group, it doesn't just go in like flat. It goes at a certain angle in space. So the more we acquire, the more we accumulate that and it turns into this spiral, this helix in space. And there are two things that were realized about the structure of DNA. Now, at the end of this class, there is a movie, which is required viewing, please. You must view it because it's examinable, but it explains a lot of this to you. Eventually, it was discovered. The DNA structure was elucidated by Watson and Crick. And this is what they found. First of all, there is a backbone on the outside made of sugar phosphate bonds, very strong. But across the interior, those bases that are on the 1' carbon actually face one another across the middle, and they match. But they match in a particular order. Wherever there is a C, a G will be present on the other strand. Wherever there is an A T will be present on the other strand. Now go back and have a look And you'll see that there are always 3 rings across the middle, okay? There's always 3 rings across the middle, look. Adenine, 1, 2, thymine, 3. Cysteine, 1, guanine, 1, 2, 1, 2, 3. 1, 2, 3. There's always 3. You always match a purine to a pyrimidine. And no matter what the order down one strand, we can always work out what is on the opposite. Just look at this little bit here that we can see, C, G, T. If I rubbed out those letters there, you could tell me what was here, G, C, A, right? Because we always match G to C, we always match C to G, we always match T to A to T. So that is what we mean. If I was to separate these strands completely from one another, I could recreate the matching strands. And that is exactly what happens during replication of the DNA. We separate the strands, we recreate. And we can recreate because of that matching rule, C to G, A to T. Okay. Those groups there, they're joined by hydrogen bonds, by weak hydrogen bonds. But the molecule is very, very stable because there are so many of those hydrogen bonds. These DNA molecules are tens of thousands of bases long. And each has its matching hydrogen bonding to its twin, its pair on the other side, which makes the molecule actually really strong. But they are easily separated. They're easily separated enzymatically during processes such as replication of the DNA.